Well, good morning. I think we're going to go ahead and get started. So good morning and also good evening to those of you joining us from Europe and elsewhere in the world. I think we have people joining us from Asia and other parts of the world. So we've got a great group of folks joining us today. I am Maury Ruffin, the Executive Director for the ARM Foundation for Cell and Gene Medicine. We are delighted that you could join us for this very important and timely discussion about the potential treatments for COVID-19 using cell therapies. The ARM Foundation is a public charity. We were formally launched in 2018 to promote education and catalyze action around issues fundamental to developing and delivering cell and gene therapies to patients who need them. We look forward to an informative presentation and dialogue today among our various speakers and presenters. And I'm delighted that we could co-host this very important webinar with the Alliance for Regenerative Medicine and ISCT. And with that, I'm going to hand it over to my friend and colleague from ARM, Janet Lambert. Janet?
Hey, thanks, Maury. Yes, let me add my welcome to Maury's. I'm Janet Lambert. I'm the CEO of the Alliance for Regenerative Medicine, which is a partner organization with the ARM Foundation. We are the advocacy international therapy sector. We have 350 members, small and large companies, research institutions, and all dedicated to trying to bring the promise of cell and gene therapy to patients around the world. So it's our pleasure to be here with Maury and also with Queenie Jiang from ISCT. And with that, I'll turn it over to Queenie.
Thank you, Janet. Good morning and good evening, everyone. And I'm Queenie Jiang, Chief Executive Officer of ISCT. As a global society, ISCT's mission is to drive the clinical translation of novel cell and gene therapies worldwide to improve patients' lives. And we welcome you all to this update on cell and gene therapy research addressing COVID-19 related diseases. This morning, I have the distinct pleasure of introducing our opening speaker, Daniel Weiss. Dr. Weiss is a pulmonologist and a critical care specialist at the University of Vermont Medical Center and a professor of medicine at the University of Vermont. He's a pioneer in cell and gene therapy research, specializing in applications for pulmonary diseases. Dan also serves as the Chief Scientific Officer of the ISCT. And in addition to him being our lead speaker this morning, Dan will also serve as our moderator for today's panel of leading experts. And so with that, I'm going to pass this on to Dan to share a bit more details about his bio and to walk us through the agenda for this morning. So on to you, Dan.
All right, thank you, Queenie, Maury, and Janet. It is a pleasure to welcome everyone to what should be a very exciting session. Cell-based therapies for COVID-19 are very timely and very important as we try and come up with ways of combating the pandemic. And so what we put together today is a distinguished panel of experts who are going to talk about the underlying science, the clinical perspectives, but also importantly, some of the ethics and business practices, as there unfortunately is a dark side to some of the cell-based therapies that are being offered out there. We appreciate that this is going to be a broad-based audience, and so we've tailored the presentations to encompass a range of scientific understanding, and we hope you all will enjoy this too. We want this to be a very interactive session, and so we encourage people to submit questions through the chat line that is available to you on the web link. And we will address the questions after each of the, and also during each of the respective sessions. So let's get going on this. So next slide, please. So as Queenie said, I'm a professor of medicine and cell biology and engineering at the University of Vermont, and the Chief Scientific Officer of the ISCT. Next slide. I do have some disclosures that I have worked with several companies on various cell-based therapy trials. I have funding from a range of nonprofit as well as industry organizations, none of which affect the content of this presentation. Next slide, please. So here's what we're going to talk about. We're going to, again, appreciating that there's a fairly broad-based audience, we're going to talk about briefly what are stem cells, because there's a still perception out there in the world that stem cell is a breadbasket umbrella type term, and that's very far from the case. There are several types of stem cells that come to public attention, and we'll work through these and see which are going to be applicable or not to their potential therapies for COVID. We'll talk at length about cell therapies in lung diseases, and in particular, something called the acute respiratory distress syndrome, or ARDS for short, as this is one of the manifestations in the severely ill COVID patients. And then we'll begin to touch on the unproven, unauthorized use of stem cells that will also be part of a subsequent part of this talk. Next slide, please. So let's briefly go through the world of stem cells. So there are three types of stem cells we're going to touch base on. The first of these are pluripotent stem cells. So the term pluripotent is what a lot of people have in mind with stem cells, in that the cell can turn into any tissue or organ of the body. And this is true to some degree. The two major ones that people work with, embryonic stem cells that are obtained, at least in the United States, from in vitro fertilization clinics, and something called induced pluripotent stem cells, or iPSCs, which are obtained actually from adult cells and have nothing to do with embryonic stem cells. And these are the cells that you see in the popular press very frequently. And these are the ones that, in that schematic picture down on the left corner, that we hope somehow we were able to manipulate, work with, and cure or treat a wide range of diseases, some of which are listed here. Everything ranging from stroke to neuromuscular dystrophy to diabetes, etc. The key thing is that at present, there is no current applicability of any of these types of cells for COVID-19 types of therapeutics. Next slide, please. The second type of stem cell that is in the public eye is the one that you've heard about that are used for things like bone marrow transplantation. And these are hematopoietic stem cells. These are cells that are obtained from the bone marrow, or you can get them from umbilical cord blood or circulating blood. And these have been widely used for years in the treatment for leukemias, lymphomas, and other types of blood cancers. And the key thing is that the cells themselves are not treating the cancer, but rather they're replenishing the bone marrow that gets destroyed during the chemotherapy or other therapy for the cancer. These have been long approved by the FDA, widely successfully utilized. And here's a definition that we're going to come back to. Autologous means that you're getting the cells from the same individual. Allogeneic means that you're going to get them from somewhere else. All right, and these are both approved for bone marrow transplantation, but again, there's no current applicability to COVID-19. Next slide, please. So, mesenchymal stem or stromal cells, these are the ones we're going to be talking about at length. And these are cells that still are not completely understood. The nomenclature, what they're called, is still in flux. But the original thinking was that these were stem cells isolated originally from bone marrow that could turn into things like cartilage, bone, fat, and what have you. This is the schematic in the upper left-hand corner. It turns out you can get these from adipose or fat tissue, placenta, umbilical cord blood, and the like too. But it turns out that their ability to differentiate, it really has nothing to do with their potential therapeutic applicability. Because as it turns out, as seen in the lower right-hand schematic, these cells secrete a variety of soluble substances. Many of these are anti-inflammatory, and these can turn off activities of inflammatory cells like T cells and B cells and other ones. And so these have been investigated for a number of years now in a fairly wide range of inflammatory and immune diseases. Things like graft-versus-host disease, things like autoimmune conditions like Crohn's disease and what have you. And they are actually approved in some countries, not yet in the United States for anything, but in some countries for a relatively small range of conditions. There's no approval as of yet for any lung disease. Next slide. So how does cell therapy with these MSCs work? Well, if you look at the schematic up there in the upper right, the MSCs are taken from somewhere in the body, be it the bone marrow or fat or placenta or cord blood, and you can either use them autologous. Here's the definition again. Autologous coming from that same individual, or allogeneic coming from somebody else. And these cells are taken out, they're put into tissue culture dishes, petri dishes, and they're grown to very large numbers. And then they're injected back into the patient with the presumptive hope of trying to treat a particular condition. Now, it's important to understand how these cells actually work. So in the lower left-hand corner, following intravenous or IV administration, the cells will lodge in the lung. They stick around in the lung. They don't turn into lung cells. They just stay in the lung for anywhere between 1 to 3 days. And while there, they will secrete a range of anti-inflammatory mediators, and hopefully that's going to do something good for the condition you're trying to treat. And then by and large, they're cleared out of the body. Or they don't stick around, or they don't stick around to any significant degree. So next slide. So how do we apply this to lung diseases? So the issue, why we do this is that many lung diseases remain incurable, and this includes critical illnesses like that acute respiratory distress syndrome that is part of the COVID disease portfolio. And it's important to understand that not all these lung diseases or critical illnesses are applicable, and it's critical to match the specific disease pathophysiology with the known biological properties of these cells. So think about these MSCs. They're injected into the bloodstream. You can actually also put them directly down into the airway. They will stick around for a few days, secrete anti-inflammatory things, mediators. Hopefully that will have a beneficial effect, and then they're gone. So where are they likely to have a good effect? So this would be in the acute inflammatory state. So a high intensity transient inflammation, something like the acute respiratory distress syndrome or sepsis or septic shock. Where they may have a possible role is in chronic inflammatory conditions like asthma or like cystic fibrosis or like bronchopulmonary dysplasia in the pediatric respiratory world, and where they're less likely to have effect. And my slide seems to be cut off on the bottom, so I'm hoping everybody can see that, are in the chronic fibrotic or destructive diseases like emphysema or pulmonary fibrosis. Next slide, please. So what is the acute respiratory distress syndrome? So this is a critical illness that results in respiratory failure, and this is what you hear about patients being in the intensive care unit on ventilators, mechanical ventilation. And there are a variety of causes that can result in the ARDS syndrome. There is unfortunately no specific treatment. We've not been able to come up with any drug or pharmaceutical despite many, many years of study. The only thing we can offer at this point is good supportive care. And unfortunately, there remains a 20 to 30% mortality. The disease or the syndrome is marked by an intense acute inflammation. There are multiple inflammatory pathways involved, and you can see in the schematic on the right, the top half is a healthy lung. The bottom half is a very inflamed lung with a lot of mediators listed that are activated during the course of this syndrome. And it causes protein leak and diffuse alveolar damage, and it's a very significant condition. Next slide, please. So if we inject an MSC into a patient with ARDS, and let's talk about before COVID, because there was active investigation into ARDS for this. There are two ways of doing this. You can give the cells intravenously, or you can actually give them directly down the airway. They will produce multiple anti-inflammatory mediators. And again, in the schematic on the right, you can see the MSC in the middle, and lots of arrows coming off with different codes for different types of anti-inflammatory mediators, all of which can have different jigsaw puzzle pieces, if you will, beneficial effects on trying to make the patient better. There are other mechanisms that are of interest to the researchers, including things like transferring mitochondria for the MSCs to cells. But the bottom line is that there is a very large literature There are many preclinical studies. So this means studies in models of lung injuries in things like rodents, small animals, large animals like pigs or sheep, or even human lungs that have been taken out of the body and used for research purposes. You can injure them and they will get better with the MSCs. So there's a solid platform for doing this in non-COVID ARDS. Next slide, please. So what about in COVID? So part of the question is whether or not the ARDS in COVID actually is as the same as ARDS not related to COVID. Many of you have heard about something called the cytokine storm, but there's evidence that this may not actually be part of the COVID pathophysiology. And one of the leading experts, Michael Maffei at UCSF, will be speaking in the next segment of this. One of the other things we think about with COVID is clotting and whether or not these patients are more prone to clot, and this will change what MSCs might be able to do. In the world of preclinical studies, all these animal studies, we're hampered by the fact that there is no good mouse model of COVID that MSCs have been studied in. There have been studies in mouse models of influenza, so a different virus, in which there have been mixed results in terms of MSCs having efficacy, but COVID still remains an open question. Next slide, please. So what's been done out there? So there have been a growing number of clinical trials pre-COVID, and we'll hear more about one of these from two of the next speakers. But most of these trials have been initial safety trials. There's limited efficacy trials, and we still don't have a good consensus on what exactly we should be doing. So MSCs can come from a variety of sources: bone marrow, placenta, umbilical cord blood, adipose, and we don't know which is going to be the best for use in critical illnesses like ARDS. The cells are similar, but not completely. They have their individual differences. We don't know if autologous, again, coming from the same person, versus allogeneic, somebody else's cells, is going to be the best way to go. We don't know if the cells are frozen and thawed, or we should keep them going in culture. This is a significant manufacturing issue for industry. We still don't have a firm sense of what the actual dose or dosing. Do we give a single dose of however many cells per patient, or do we give multiple doses? If we give multiple doses, over what time frame? These are still open questions. And again, we're not quite sure if intravenous or systemic versus airway, or even injecting the cells directly into the muscles might be a way to go. Next slide, please. So there is compelling, again, preclinical data in animal models, and this has led, again, pre-COVID, to several trials, one of which we'll hear again a little bit more, that was multi-center, randomized, double-blinded, placebo-controlled trial sponsored by the National Institutes of Health in the US, coordinated out of the University of California in San Francisco, in which allogeneic, so someone else's cells, were given, and these are bone marrow in origin, as a single IV infusion to patients with ARDS. Now, the thing we're always worried about is safety. And so the primary outcome for the initial studies showed that this was a safe approach. There were no adverse effects, no obvious downsides. That's critically important, so it lets the field move ahead. And so a phase 2, an efficacy trial, was completed and published last year. And again, safety was achieved, but we didn't quite see the type of efficacy that we wanted to see, and there are potential reasons for this, and we're working to overcome these and hopefully make this work correctly. Next slide, please. So, COVID. COVID is a scary disease, particularly for critically ill patients. And one of the things that we've learned is that patients tend to have the propensity to form blood clots throughout the body, and some of the systems involved in clotting seem to be deranged. And so this raises a possibility that MSCs, if they're injected into the bloodstream, might clot in lungs of COVID patients. We haven't seen that in any non-COVID patients as of yet, but it's an open concern and something we need to pay attention to in the COVID patients. Next slide, please. So one thing I also want to throw at you, we're talking about cells, but it turns out that cells secrete lots of little particles, and you may have heard about these. They're called variably extracellular vesicles, or exosomes, and you can see in the schematic on the right side these small little blue buds that come off of the cells. And these exosomes contain genetic materials and mitochondria and other things that potentially could convey the same type of benefit at the cells themselves. So there is a growing preclinical, again, studies in animals showing that the exosomes themselves may be as effective as the cells. We're just beginning to see some clinical investigations on this. And there's also a possibility that these exosomes could be actually aerosolized so that you could breathe them in. But this is all very premature, speculative, and we do not have any proven therapies as of yet with exosomes. Next slide, please. Next slide, please. So where are we at with COVID? In the last few minutes of the scientific background, the field is moving quickly. So this is admittedly a little bit old, but as of May of 2020, there were 54 trials listed on the clinicaltrials.gov website. And these show the geographic distribution. Many of these are in China, many of these in the United States. And off to the right is a small figure showing the types of cells, the origin of the MSCs that were utilized in these different studies. Next slide, please. All right, what do you do in a pandemic? So desperate, compassionate use, we understand that, we support that. People are dying and you need to do something. And so the initial investigations for many of these COVID trials of cell-based therapy were observational. Patients got the cells, everybody knew they were getting the cells, and hopefully they would get better. But you cannot, if they got better or not, you cannot determine any causality. And so what's needed is a randomized, double-blind, placebo-controlled trial that gives full transparency on the investigational product, quality control, outcome measures according to standard guidelines, and data available for independent analyses. And we're going to talk about all of this in a subsequent portion of this session. We also strongly advocate that whenever possible that you try and ask mechanistic questions in the context of a clinical trial. Can you learn something about how these cells actually are working? Next slide, please. So the last slide I will leave you with is this is all good and forward-looking, but we have a problem. And this is something called stem cell medical tourism, or phrased a different way, direct-to-consumer stem cell innovations. And many of you are familiar with this, and the picture on the right kind of says it all. There's a worldwide proliferation of clinics and other outfits offering unproven therapies for a range of diseases, including lung diseases, including now COVID ARDS. This clearly takes advantage of desperate patients, and these approaches are potentially dangerous. And it's exacerbated by the fact that there are different regulatory frameworks across different countries. This is a problem with COVID. We're going to talk about it more at length, but last slide, next slide, please, is just to give you some basic definitions. How do you define an unproven cell-based therapy? First, there's an unclear scientific rationale to suggest efficacy. There's a lack of understanding of scientific mechanism and or biologic function to support clinical use. Insufficient data from laboratory studies, animal models, or clinical studies. Lack of a standardized approach for product quality or manufacturing consistency. Inadequate information disclosed to patients for informed consent. This is a critical, serious issues. Use of non-standardized or non-validated methods of administration, and uncontrolled experimental procedures in humans. So with this, I want to thank a lot of people in my lab and my world who have helped make this happen. And what we're going to do, go to the next slide, is we're going to move immediately to introducing our next speakers, Carolyn Hendrickson and Michael Mathey, both pulmonary and critical care physicians at UCSF, who are going to lead us through clinical perspectives. And just to say a brief word about both of them, Dr. Hendrickson is assistant professor of medicine at UCSF, clinical trial physician for the San Francisco General Hospital and Trauma Center. And Michael Mathey is a world-renowned expert in ARDS, professor of pulmonary critical care allergy and sleep medicine at UCSF. They will take over now, but what we will do is weave the various questions I've seen coming in on chat into the conversation with Drs. Hendrickson and Mathey, and we'll hope to get to all of your questions. So Michael and Carolyn, please take it away.
Thanks very much, Dan. That was a terrific overview, covered so many of the key issues. I think my colleague, Dr. Hendrickson, is also on the line.
Yeah,
or maybe
I'm
not.
here.
Yes, she's right there. Good. Hi, Carolyn. Carolyn's been busy in the ICU already this morning. I was just going to give a very short follow-up to the remarks that Dan gave regarding our clinical trial. We have this phase 2B randomized double-blind clinical trial of allogeneic bone marrow-derived mesenchymal stromal cells supplied by the University of Minnesota. It's being conducted at 5 major university centers and 6 hospitals. We really just got started in January, and this is intended to test efficacy in 120 patients, with the primary endpoint being improvement in respiratory function by an index we call oxygenation index, a combination of airway pressure and oxygenation. And then with the onset of COVID in February and March, we had a large number of patients here in San Francisco. And Carolyn Hendrickson, is the site principal investigator at San Francisco General, actually enrolled a large number of patients throughout the spring in this trial. And I'm going to let her elaborate a little bit more on that trial and what was involved. And you might say a word, Carolyn, about what you did to try and maintain safety among the healthcare workers and the investigators.
Sure. So we enrolled patients with COVID ARDS in a trial that was designed to look at ARDS more broadly, and had to adapt our protocols for safety and personal protective equipment conservation. So we got creative with how we administered our study drug. The protocol calls for the study drug, which is either cells or placebo, and it's blinded. So we receive a product from the bone marrow transplant lab that has been sort of cloaked in opaque plastic. But for the COVID ICU patients, we actually run these very long lines of tubing from the patient's bedside to outside the hospital room so that nurses can titrate medications through the pumps without going in to be exposed at the bedside, and also without using their PPE for these frequent adjustments of infusions. So we had to hang our study drug outside also because we couldn't justify using PPE that was in short supply for a clinical trial while people were worried about their safety and providing direct care to the patients. And so I would sit at the bedside and push, a few cc's every minute, like a very fancy IV pump through this tubing until we figured out a way to run a device that uses a pressure bag and a drop counter to get the correct infusion rate over time. I guess I would also say the major difference that I observed in running a clinical trial in this environment is that patients and families were very generous with their willingness to participate. With an emerging disease without proven therapies, people were eager to access a promising and safe therapy, and were more agreeable than I have ever encountered in other consenting processes. So we have learned a lot already and will continue to learn from this experience.
So Carolyn, the last comments are of course apropos in a urgent emergent pandemic situation. Better nature comes out of people and the like in circumstances. But I want to go back, I want to pull out one of the chat questions here that's come through for both you and Dr. Mathey. Reading verbatim from the question, Dr. Aswin Hari. Oh, that's me. What is the logic? What is the logic behind using cells in general for the treatment of the ARDS, especially COVID-related when there's so much going on in the lung and you have many neutrophils and other cells stuck in capillaries, if the cells are given IV, is it reasonable to expect many of these cells to survive in the blood to get to the target tissue and deliver? So the question is really, it's a broad-based question. Why are we using cells for ARDS? And why, how would that be even more or less or equally applicable for COVID-associated ARDS?
Well, Carol and I can both answer that. I'll start by saying that our preclinical studies in small animals and large animals, including a sheep model, as well as an ex vivo perfused human lung model. We studied lungs that have been rejected for transplant or they weren't used, but most of those human lungs come to our laboratory are in reasonably good shape. And so in the human lungs, we simulate pneumonia with bacteria, and in the large and small animal models, we've simulated ARDS, including, as Dan alluded to, viral pneumonias in mice. And the two major potential areas of efficacy for the MSCs is they reduce the acute injury and inflammation. That's what we saw in the preclinical models. And then there's several lines of evidence from longer preclinical models that they may enhance the repair process. They may turn the inflammatory environment to being more of a pro-repair environment in terms of how the macrophages, for example, are programmed. And they do have antibacterial effects. As Dan alluded to, we have mixed results with viral injuries, though one model of H5N1 influenza lung injury showed a definite benefit in terms of survival in older mice. But we don't know in terms of SARS-CoV-2 whether or not there'll be a specific therapy, that therapeutic effect. Although with the presence of an antiviral that works, remdesivir, my view is that there's an additional opportunity for ancillary therapies such as cell-based therapies. But what else would you say, Carolyn, in terms of the rationale or related issues?
I would just add a framework for thinking about this. And when you're trying to develop a treatment for a very heterogeneous disease, it is appealing to use something that's a smart therapy that may sense and adapt to its environment and work through multiple simultaneous mechanisms that are appropriate to whatever the microenvironment the cell is seeing. And the idea that we're going to find one protein that sort of is the key to solving ARDS and target that with a monoclonal antibody makes very little sense to me in a disease that's as heterogeneous as this is.
So I think you could encapsulate both of what you've both said. There's a solid biological and mechanistic base for considering these cells in this acute high-intensity inflammatory situation. We still don't understand the full range of the anti-inflammatory portfolio that they exhibit, but it does make sense in a disease that has multiple inflammatory pathways. And there's a solid platform, again, of preclinical studies in animals and the like that shows that these really is a good possibility. So let me broaden this out. There are a couple more questions that have come through. So I'm going to put a few of them together. So this is ARDS related to COVID. And do you think that any observations in this scenario, in this setting, will be applicable to ARDS? Again, ARDS results from a variety of different etiologies. Pre-COVID, we had ARDS, and post-COVID, we'll still have ARDS. Will the results be applicable to non-COVID ARDS? And then also, too, have you seen any evidence that there's been any clotting problem in those patients that you've, COVID patients, that you've administered cells to as of yet?
I don't think I can answer the really important question, which is how different is COVID ARDS from other forms? As an emerging disease that hasn't even been around for a year, we have really limited understanding of its full breadth. and the full spectrum of respiratory failure in these patients. Why some people get it, why others don't, why it's really severe in some and much more mild in others. So I have no idea if it's going to be broadly applicable or not, the therapies that we develop for COVID ARDS. But again, I would just touch back on the comment I made earlier that if these cells have the possibility of acting through a variety of mechanisms, perhaps they're able to target different phenotypes of the same syndrome. And with regards to seeing clotting in patients who have been administered MSCs, so I'm blind to who has gotten cells and who hasn't. We've found of the adverse events that we've reported to our DSMB, none have been attributable to study drug. And so I think the answer is not that I know of. And we have certainly seen lots of clotting outside of the study. And so I
Right.
would be dubious of attributing any of the thromboembolic complications to a one-time infusion of a study drug.
So I would add to that also too, there in parallel with your trial, which is an academic-based trial, there are several industry trials currently ongoing globally, but the ones in the United States that I'm aware of have not shown or presented at least any evidence of significant clotting issues related to the MSCs being administered to COVID-19 patients. But again, we have not yet seen the full scope of data from any of these trials. And so again, this remains a theoretical concern, but one that we all want to keep a sharp eye on. So to go to a couple of the other questions that were here. So we've made the point there is no preclinical model, mouse model as of yet for COVID-related lung injury. There are transgenic mice, and there are also primates who apparently can be infected with COVID, and I think ferrets as well too. So one of the questions is, well, who cares? Do we really need a preclinical model? Why not just full steam ahead with the clinical investigation? So this is a kind of a broader based philosophical question. What do you think?
Well, I would say briefly that because of the lack of understanding, as Carolyn said, about the features of COVID-19 ARDS that are different from regular ARDS, as well as the overlap features that are similar, we really cannot at this point depend on preclinical data to guide us. I think that the clinical syndrome has already been studied, and we'll know a lot more in several months, but I think what's happened is inevitable that many different therapies are in the process of being evaluated. The one therapy that has shown benefit unequivocally is remdesivir, the antiviral, when given to hospitalized patients who required, who were sick with COVID-19. And there are several other trials in process, including plans to study monoclonal antibodies, their convalescent plasma studies. And there have been several monoclonal antibody studies designed to inhibit, for example, interleukin-6. None of those have yet panned out, although some of the early trials did not include remdesivir. So that limits interpretation of that data. But I think the cell-based therapies are reasonable rationale because there are multiple potential benefits for reducing injury and enhancing repair. Maybe in time there'll be preclinical models to help us in this area. And one area of interest that Dan mentioned is the whole area of extracellular vesicles. And that, of course, is an offshoot, if you will, of MSC therapy. And that might be amenable to some preclinical studies eventually with SARS-CoV-2. But right now, we just have to use our best judgment of which therapies are reasonable to test, which have a low risk of harm.
Very good. So I see, Carolyn, you're beginning to reply through chat to some of the questions. And so we won't get to all the questions. We'll try verbally for open discussion, but we will try and respond to as many as possible. There is a question on airway delivery of cells. So we appreciate that cells being administered, there are a lot of good reasons for them. There are other therapeutics that are being investigated, as you said, remdesivir and the like. And, but if we're keeping our focus on MSCs, so one of the things that we've begun to see is, can you use the airways? So can you put cells directly down the airways and have the same type of effects? And arguably that would avoid potential clotting problems and things of that nature. So Michael, I know you've had experience and advocated for this in the past. What is your thinking at present on airway delivery?
Well, I think it's a very interesting idea. When we first proposed that as a possibility with the Federal Drug Administration, FDA said that they thought it was too complicated because to try to deliver the cells uniformly to the adult lung would have required almost a fiber optic bronchoscopy delivery to the 19 different segments of both lungs. And we had actually, in our first preclinical study, found efficacy in the human lung by delivering the cells directly into the distal air spaces. Those subsequent studies showed we had the same effect by delivering them in the perfusate, the intravenous route. I do think that the exosome microvesicle approach that Dan touched on might be more amenable to airway delivery or delivery by an aerosol. And that is, several groups are interested in that as a possible strategy. Even in the neonatal setting, Dr. Kurumbanis in Boston is studying exosomes from MSCs for treatment of premature lung disease in human infants. So cell-based therapy might transform to a delivery via the air spaces, particularly if it were with extracellular vesicles.
No, that makes a lot of sense, but I want to stress a few things with respect to that, because we're already seeing out there advertisements for aerosol delivery of exosomes, and including do-it-yourself at home advertisements. And it's critical to understand that while a very intriguing approach, we simply do not have data to show that this is going to work. And in particular, we don't have the type of data to say that if you have an exosome, and again, these are these small little particles produced by cells, that if they're breathed in or administered by a nebulizer or some other device, that they're actually going to get to where you want. But more importantly, the ARDS lung is a very inflammatory environment. And there are lots of things that could potentially block the exosomes, destroy the exosomes, as you're trying to get them to where you want to get them into the lung. And so these are the types of scientific platforms that we need to develop and show can work on the way to developing an aerosolized or aerosol-based type of therapy. So I fully agree this is a ripe area but for investigation, but I want to make sure nobody on this participating in this session walks away with the wrong message that this is established therapy. So, all right, so we have a few more minutes before we move on to the next portion of this presentation. And there are a number of questions that we will respond to accordingly through the chat. But to Drs. Hendrickson and M.P.H, what else would you add to this discussion for cell-based therapies for COVID ARDS at this point? Is there anything that's important for people to walk away with that we've not yet covered?
Well, I see one area here where there are questions. Carolyn may have been responding, but there are questions about vitamin C and also dexamethasone. And of course, both of these represent other potential therapies. Vitamin C had a very positive phase 2 trial pre-COVID in ARDS patients published in JAMA. It still is possible that vitamin C high dose might be useful to test in this COVID-19 ARDS environment. It is a candidate, but not yet targeted in a trial. Dexamethasone, I'm going to let Carolyn comment on from the recovery trial.
I'll keep my comments short because I have not seen the primary data from that trial yet. But the effect size that is reported in the preprint is staggering. And I'm anxiously awaiting more information about how those subjects were selected from the population that gave rise to all of the cases they were treating, and to see a little bit more information. I will say that I have some concerns about using like a moderate dose of steroid for 10 days in a lot of patients who we've seen get complications of respiratory infections, ventilator-associated pneumonia, as well as a lot of delirium and weakness. And so I'm not sure that the study is going to answer questions that we have about the longer-term deleterious effects, but like I said, the mortality benefit they are reporting is pretty astounding. I do think that there is still room for improvement, even if remdesivir and dexamethasone become the backbone of the therapies that we're studying.
Right. So it brings up another important point, is that there are several potential therapies being investigated across the board. Antivirals, anti-inflammatories like dexamethasone, cell-based therapies. None of these are mutually exclusive. And in many cases, critically ill patients may be on multiple of these experimental therapies because that's just what we need to do at this point. So to tease out specific benefits in combined trials, if you will, may be a little bit difficult. But the last question I want to have you consider is actually one we haven't spoken about, and it just came in. When do you start with the MSC treatment? When the patient, what's the best? And I'm using that term deliberately because there is no best. Do you start when they're already critically ill on the ventilator? Do you start when they are admitted to the hospital, when they're looking like they're going to deteriorate, et cetera? What's your best operating principle right now for that?
Carolyn, why don't you explain how we approach it in our trials, safety and our efficacy trial?
Sure. For the trial, we're enrolling patients who are early in their ARDS course. I think you raise a really excellent question, which is how early is early? We do think that patients, have substantial lung injury before they make it to the ICU and are intubated, but that's not how our study is designed. We are actually testing patients who have been intubated. They're in the ICU for less than 120 hours and qualifying for ARDS for 72 hours. Or less. I mean, the sooner the better. We're basically screening all the time and enrolling them as soon as their PaO2 to FiO2 ratio, which is a gauge of the severity of hypoxemia, is less than 200. And our aim is always to enroll as early as possible in that window.
Dan, can I have one more small response to a question
Yes, of course.
asked? I just want to make it clear that the MSCs that we use in this trial, these clinical grade cells produced by the NIH repository at the University of Minnesota, we tested these precise cells in all of our preclinical models, including the human lung and the sheep studies, and also in our safety trials, as Dan mentioned, in humans. And yes, these cells do produce IL-6 and IL-8 and other pro-inflammatory factors. This is true. Although when we measured the levels in our phase 2A trial, there was no difference in the levels of IL-6 or IL-8 in the first 2 days. We did find a reduction in angiopoietin-2, a marker and mediator of endothelial injury, and that was encouraging for a biologic effect. but I just wanted to be responsive to that question.
So I want to, one brief comment. I see, looking that there's people who are entering questions on the Q&A. And so if possible, if you could enter them onto the chat as opposed to Q&A, then that would be visible to everyone and give broader visibility. But I do want to leave this with one combined question here. So there's a commentary about the pace of clinical trials in this pandemic situation. Are we having enough trials? Do we need more trials? And I'm going to combine this with a comment made separately in the context of BARDA, the governmental agency, essentially removing their support for a number of trials, including cell-based therapy trials, in favor of focusing entirely on vaccines. So kind of a broad-based question, are we doing enough that we should be, or should we be doing even more cell-based therapy trials at this point?
Well, we're just writing a short review article, and it seems like there are several cell-based therapy trials, Dan, you probably know more than I do on this, internationally, that are registered on clinicaltrials.gov. I think the main issue is doing high-quality trials, blinded and randomized. That's the benefit of the remdesivir trial. It was blinded, randomized, well-designed. That's true of our hydroxychloroquine trial that was blinded and randomized and showed no benefit. And so I'm concerned about open label trials when they're not appropriate controls. That's one of the issues that will be discussed a lot when the dexamethasone trial is published from the recovery pragmatic approach. So I don't know if we need more. I think we need well-designed trials. I do think there's room for a trial which includes giving MSCs twice, once like we are doing at the early part of ARDS and potentially a second dose 2 to 3 days later if the patient is still being ventilated. So I favor that as a way to try and more completely test efficacy. But I'm most concerned that the trial design be optimal.
So that's an excellent point to leave off with. There are many trials out there that you can find listed, but not all of them are necessarily being done in a way that is best going to yield the type of useful information that we need to decide whether or not the treatment was effective. There are some of these trials actually have multiple dosings, and there's trials up to 4 separate dosings. And so it's a bit all over the map. What we do want to do is, if you could go to the next slide, please, is let's move on to the final portion of this webinar, if you will. And we want to talk about the ethics and business practices. And so to do this, we have, again, another distinguished panel of experts, Laerdis Economou, who is an associate professor in the Department of Oral Biology at the University of Buffalo, SUNY Buffalo. Michael Lemke, who's Director of Scientific and Industry Affairs at the Alliance for Regenerative Medicine, and Patricia Zettler, who's an Associate Professor of Law at the Ohio State University Moritz College of Law, who are going to lead us through the considerations about how to best do this and the unfortunately unethical things that are out there. So please take it away.
Thank you, Dan. I think I can start. So as Dan mentioned, I'm the current chair of the ISCT PTF on the use of unproven and oral ethical cell and gene therapy. And I'll give you just a brief overview of the PTF. It was founded in 2014, and it was used to call the PTF on the use of unproven cellular therapy. So the new title, I think, reflects the expanded repertoire of these unproven interventions, both cell and gene-based. And we have a lot of experts in regulatory science, basic research in stem cell biology, bioethics, public policy, and cell and gene therapies. So our main focus is to characterize unproven cell and gene interventions and also promote safe and effective treatments worldwide. And I think the key word there is worldwide, because we are faced with a global borderless challenge that is posed by this industry, the direct-to-consumer industry of unproven interventions. So this industry emerged more than 10 years ago, 10 to 15 years ago. It has been continuously transforming, reinventing itself. But the common thing there, the common thread is it offers unproven, unlicensed, and potentially dangerous treatments. And these treatments can come from various sources. The material that's been injected or administered to patients can be stromal vascular fraction from fat tissue. Increasingly, we see perinatal tissue products such as umbilical cord, placental products, but there's been also bone marrow. And lately, of course, we see the emergence of acellular products such as exosomes. So I want to be very clear here. Independently of the source of the product, I think that what is really sold here to the patients is one thing, and it's false hope, nothing more than that. So that's a problem. This industry is, I would say, an obstacle in the legitimate development of cell and gene-based interventions. And it's not surprising that with the COVID-19 pandemic, we see this industry now offering similar treatments for either what they call prevention of or even cure of COVID-19. And we see increasing advertising of these treatments, increasingly administration or promotion of exosome treatments. And it's in the context of what they call preventive of prophylactic measures. So it was nicely pointed by Lee Turner recently in a Cell Stem Cell paper. These businesses have to be very careful with their claims because we have a very serious public health issue. So they have to be careful not to draw the attention of the regulatory agencies. So what we see there is a lot of vague advertising claims that have to do even borderline about wellness. We see claims about healing properties of their treatments, boosting of the immune system, and improvement of overall health. We think it's understandable. This is still problematic for both scientific and ethical reasons. And fortunately, there's been backlash, there's been reaction to these claims. The reaction comes both from scientific and professional organizations and the regulatory agencies. On the front of the professional and scientific organizations, we've seen very strongly worded statements about these treatments, statements that come from various organizations, including the ISCT and the ARM, that make it very clear that no proven or approved treatments, cell-based or gene-based treatments for COVID-19. And it's obvious also that the regulatory agencies have taken note of the situation. I counted within the 11 letters of the FDA, the 11 letters that the FDA issued to businesses offering cell-based products or exosomes for 2020. I counted 5 letters that mention these claims about COVID-19. And also the FTC has issued at least 5 to 6 letters in these businesses with very strongly worded letters that make it very clear that these claims are unfounded and amount to deceptive advertising. So the stance, I would say, of the regulatory agencies and professional and scientific organizations is very strong. But still also part of the discussion that will follow how we deal with the situation.
Thanks, Leonidas. Maybe I'll just take the microphone next. My name is Michael Lemke. I'm with the Alliance for Regenerative Medicine, the Director of Science and Industry Affairs there. And I'm going to try to, as best I can, represent the perspective of therapeutic developers. As Janet Lambert mentioned at the top of the webinar, ARM has over 350 member organizations, the majority of whom are therapeutic developers, cell and gene therapies and tissue engineered products, including several who are currently working on cell therapy treatments for COVID-19 and complications related to COVID-19. I want to echo something Leonidas said, which is that there are currently none of these therapies are approved, and that's an important takeaway for today. They're not approved yet. although our members are, according to the code of conduct that all of our members sign, going about seeking approval for these therapies in the appropriate way. And we think that's very important. We maintain an open relationship with the FDA and the EMA. We try to advocate with them for clear, predictable, and efficient regulatory pathways. We believe that the FDA has been very responsive in this situation of COVID-19 and acted as quickly as they're able to approve trials or studies of experimental drugs that are submitted to them and have the appropriate clinical trial designs. Other than that, I would just say that we, as Lyra's pointed out, we do not advocate for fewer regulations or no regulations. We want these therapies to come in the market with safety and efficacy demonstrated. And we continue to work with groups like ISCT and the ARM Foundation towards that goal.
So I guess I'm going last on our panel. As Dan mentioned, I'm a law professor and a lawyer by training. I'm a former FDA attorney, and now I'm an associate professor of law at The Ohio State University and a member of Ohio State's Comprehensive Cancer Center and Drug Enforcement and Policy Center. I'm going to start with the caveat I always give as a lawyer by training, which is that although I am a lawyer, I am not your lawyer. So everything I say today is for educational purposes only. So Mike and Laird just offered some, I think, helpful thoughts about business models, marketing, unproven interventions for COVID-19 and ethical concerns around those kinds of business practices. I think I want to sort of emphasize two things. One, as Mike mentioned, right, nothing is approved right now, and nothing is FDA approved right now, and that's true even for things like remdesivir, for which there are good data supporting them. In times of public health emergency, FDA has the authority to issue something known as an emergency use authorization. And the standard for an emergency use authorization is not substantial evidence of effectiveness, the sort of typical 3 phases of clinical trials that we think about, but rather it's reasonable to believe that the intervention or the technology may be effective for the relevant condition. So FDA issued EUAs for hydroxychloroquine and chloroquine back in March and has since revoked them, but the data supporting those were not nearly as good as that for remdesivir, to say the least. And you can sort of understand, though, hearing that standard, reasonable to believe it may be effective, why those EUAs could have been issued in the first place. And I think it's helpful to understand that nothing is FDA approved as of now, and those are all, those emergency use authorizations or EUAs, we can think about them as forms of pre-approval access. I guess I also want to echo a comment from the previous panel about sort of the importance of getting good data from randomized, controlled, double-blind, blinded trials, right? So just in thinking about the role of the FDA and why we have FDA approval, even in these kinds of urgent public health emergency situations, we often think about FDA as protecting us physically from unsafe and ineffective products. But another way to think about FDA approval is that through requiring approval from FDA or similar regulatory bodies, we are requiring companies that want to market products to produce that kind of rigorous evidence that healthcare professionals and patients need in order to make decisions about clinical care. So FDA has this sort of traditional consumer protection role of protecting us from unsafe and ineffective things, but also this information producing role. So it's encouraging innovation, right? We don't want new things for new things' sake. We want new interventions that actually work. It's not innovation if it doesn't work, right? So I think that's one helpful way to think about what FDA's role can be here and why it's important that we have the FDA approval process, even in these sorts of urgent public health, and maybe especially in these sorts of urgent public health times.
Let me throw in a comment on to that, and then been reading through the chat and QA questions. There's a sense that why are we waiting? Why aren't we pushing ahead further and faster and the like? It's a pandemic. It's etc. And the response, I think you articulated very well. We need assurance, number one, primarily that any approach is going to be safe and do no harm. Secondly, we need assurance that it's going to work, right? And you can't get assurances that something's going to work unless a investigation is done in a way that's going to yield that information in an unequivocal way. And so it's certainly good, again, pandemic situation, go ahead, throw the kitchen sink. So go ahead and give patients cells, for example, of whatever type. And there were several industry reports just on that. We had 12 patients that got cells and they seemed to get better. But again, that's not definitive data. They might have gotten better anyway. We don't know. But at least they showed initial safety. And that gives us a platform, again, even in a pandemic situation, where we should then push ahead with the proper type of evaluation to determine if there truly is efficacy. Beyond that, it's a rush to false judgment to assume that we should do things any other way at this point.
Yeah, I see a question in the chat about EAPs or expanded access programs and sort of how they're thought of. And I think so those of us that are sort of familiar with expanded access programs or compassionate use or special access programs in Canada or, right to try, I think it's useful to understand emergency use authorization sort of in that pre-approval access context, as we already recognize outside of public health emergencies that there can be times where individual patients are willing to take on more uncertainty or more risk and access products before we have all of the evidence. And so we can think about emergency use authorizations like that, I think, but there's this, we have to be mindful, and this is something that's in FDA's expanded access regulations, we have to be mindful of not allowing such access to delay or halt sort of the development of the data we need to really understand whether the interventions work.
So
yeah, I think.
Yeah, so Mike,
Sorry,
let me throw something towards the three of you. What more should we be doing to make this happen properly? And there's the positive side to do proper clinical trials. There's the dark side, which is how do we, and I'll be quite brusque about this, wipe the snake oil off the face of the map? What else could the FDA, other governmental agencies be doing at this point?
Yeah, I can answer that. I can start with the answer. So I think, well, we may not be able to totally eliminate from the face of the earth, because it comes, unfortunately, our field is a victim of its own success. So as we have more promising cell and gene-based treatments coming to the clinic, we'll always have this parallel industry that will offer this type of snake oil, and they are very adept in advertising this, so selling this. But I think the regulatory agencies have a very big role to play in this aspect. So we can educate, like the ISCT, PTF can educate the public, we can inform, we can write papers, but if the regulatory agencies do not take a more forceful stand, and they should start enforcing their own regulations. And I think this is happening increasingly. But I think the most important player in this equation will be a regulatory agency, such as the FDA. Enforcement, and make sure that a lot of these businesses do not get into this arena. Because I think a lot of these players are just opportunistic. They try to find where the bar is, what the regulatory obstacles are, and then they just get into there. There is no real commitment to any type of legitimate cell or gene therapy.
Yeah, and I would maybe just add that something I think expanded access can help if it's done in the right way. And as Patty said, the FDA does have regulations that cover that. It can be done in the context of an appropriately designed clinical trial. And that is in fact being done now in this situation in some cases. So we should support the FDA's efforts to allow that, and hopefully they will continue to do so in the appropriate way. Again, in the context, I think, of controlled clinical trials. And another important point is if people are receiving unproven therapies, they're potentially removing themselves from being candidates for clinical trials for therapies that are being studied in the right way. And one problem we have now is just to be able to enroll enough patients quickly enough to get the evidence that these therapies are safe and that they work.
Yeah, I'll just add one comment that FDA and FTC, the Federal Trade Commission, have announced that fraudulent COVID-19, things fraudulently marketed for COVID-19 without supporting data are an enforcement priority. And so it may be, even though, there's sort of a history of FDA being a little lax, at least, several years ago with respect to enforcement, it may be that there's enforcement, but it's never possible to 100% enforce regulations.
All right, very good. So any other thoughts the three of you want to throw into the mix? We want good trials. We want trials that are going to be conducted in a way that are going to give us the type of answer and information that we want. Sometimes trials don't work, even if they're conducted as best possible. Everyone acknowledges that, of course, but you cannot not do a good trial. We cannot rely on bad trials. And then we certainly will do everything possible to diminish the influence of the unproven cell therapy industry, particularly for COVID. So as we get into the final few minutes of this presentation and webinar, thoughts from the three of you? Try and tie this all together.
Yeah, I would, I guess the final thought that I have, Dan, is I think, we do support the FDA's enforcement actions and enforcement discretion, but, we all know they're resource constrained. And I think we all the stakeholders and many of the people, if not all the people listening to the webinar today, have a responsibility to, celebrate and point out the good science and the well-designed trials, but also to call out when people are selling false hope or promoting stem cell therapies inappropriately.
All right, very good.
And I would like to add to that we also should, so legitimate developers of cell and gene therapy should be very careful about hyping, because that's been a problem also in basic science, hyping stem cell therapies. That feeds into the discourse of this type of businesses. So you have to be very clear about the limitations, about the benefits of cell-based therapies, and they're always condition specific, and also about the timelines when we can get a cell therapy that is being proven safe and effective into to the clinic and to patients.
All right, very good. So I'm going to just in the final minutes, there's some last minute questions that have come in that I think are worth considering even briefly. And the one I'll focus on in this very last minute, the cost benefit. Does the cost of a new therapy, MSC based, merit its use? And I think the answer is we don't know. We can certainly calculate the cost of patient care in the ICU, lost productivity, lost societal costs, all those type of things, versus the cost of an MSC. And it's a fair question, and one that will be on everyone's mind, I think, as we proceed forward once we show efficacy. So we're at the end of this webinar, and I want to thank very gratefully all the participants for wonderful presentations, discussions, considerations. We want to thank all of the participants who listened in, and particularly those who've asked questions. We hope we've answered most of them. I realize there's some nitty-gritty questions on the biology of the MSCs we didn't address as a group. We will try and get to those through the chat response. But again, we thank you all, and we want to thank ARM, the ARM Foundation, and ISCT for putting together and sponsoring this webinar. We wish you all to stay safe, stay healthy, and thank you for participating. Thank you, everyone.
Thank you, Dan.
Thanks, Dan.